Hi, I'm Charlie Casper, and I am going to talk about randomized trials and give you an introduction to that. As far as objectives, at the end of this module, we hope that students will be able to first understand the rationale for randomization and also its advantages. We hope you will be able to recognize pitfalls associated with missing data and how that relates to randomization. And also, you should understand the concepts of non-adherence and intention to treat analysis. So this is really just going to be a brief overview, and you can imagine that there are entire books and courses that are devoted to this particular topic, and so it's impossible for us to cover everything or to really go into great detail on any of these topics in particular. But maybe this information will be able to give you some background on clinical trials and why we do them, some of the terminology involved, and some of the big issues that we run into. Here's an outline of the topics which are related to the objectives. So we'll first talk about causation and how that relates to randomization and what the real rationale for randomizing is. We'll then talk about the specifics of how you might randomize. And then we'll talk about missing data and how that relates back to randomization. Then we'll talk about a problem in almost every clinical trial, and that's non-adherence, and how that then ties into the intention to treat analysis. So what is the purpose of a clinical trial? From where I stand, the purpose is to establish whether treatment A is superior to treatment B. Now, there are different kinds of designs that may involve more treatments and may, involve a different structure than this, but this is the basic idea behind a clinical trial. Is the new treatment superior to control, for example? And we allow a certain level of statistical uncertainty. You may have heard of alpha. That's what we're talking about. It's this margin that we allow, the margin of error. So beyond that statistical uncertainty, the investigator really has to limit the impact of the random variation, for example, by having a large enough sample size, and they should eliminate, to the greatest extent possible, all other sources of uncertainty besides the statistical. And any other remaining uncertainty can actually diminish the validity of the trial or undermine it entirely. Well, let's talk about statistics for a minute. We're not going to go into too much detail on statistics, but the basic idea is that you have a lot of people in your trial. People are not all the same. This leads to what we might call random variation in outcomes. And when you're comparing two groups to each other, we may consider the difference between the two groups to be statistically significant if the difference in, say, averages is large compared to the standard deviation. Now, there are different kinds of outcomes and such, but the basic idea is this. If the differences are large compared to the variability, then we claim statistical significance. Now, statistics is great and can tell us whether two groups are different, but not why. So, for example, if I have two groups and one has sicker patients than the other group, then statistics may tell us, in fact, that the groups are different and that the first group has worse outcomes, but it's not going to tell us why the groups are different, which in this case is because they are sicker. We rely on outside information to determine why the groups are different. So now let's get to causation. This is really what's behind randomized trials. So first, what are we talking about? So if we say X causes Y, that means that Y would be different if X were different. Stated another way, it's all about what would have happened if. Okay? Now, if we could only have the power of omniscience, we would know what would happen if a trial subject receives treatment A or treatment B. And with that information, we would then know the causal effect of treatment A compared to treatment B for that particular subject. However, for a given trial subject, we can never know what would have happened if the subject received the other treatment. We only get to observe what happens to them with the treatment that they are given. So, from this point of view, things seem a little bit futile, and apparently it's impossible to truly ascertain causation. Now, what if we had a population of sets of perfect twins? Now, I should say perfect twins don't just mean identical twins, because there are lots of identical twins out there in the world, but even identical twins have very small differences, and ultimately we don't know whether their outcomes would be identical. But anyway, these are perfect twins. Perfect in every way, including what outcome they would experience on either treatment. In that case, we could give one twin treatment A and the other treatment B. And if we do this for every single twin pair in our trial, then that's going to be just as good as being omniscient, because we could know the causal effect of treatment A versus treatment B for any of these individual twins, and then combine that information to get a population assessment of the effect of treatment. However, as you may have guessed, there are no perfect twins in the world, so we are again stuck. How can we get at this causation idea? Which brings us to why randomize. Now, the typical rationale that you may have heard for randomization may go something like this. Randomization tends to produce comparable groups with respect to risk factors, both known and unknown, remove investigator bias in allocation, and guarantee that statistical tests will be valid. And you'll find these in a lot of very respected clinical trials texts and other sources. And I'm not saying that they're wrong, they are largely true, but these have nothing to do with causation. The true purpose of randomization is to provide random subsamples of the potential outcomes, in other words, the what-ifs of the trial subjects, in order to simulate a population of perfect twins. That's exactly what randomization is doing. And in that same way, randomization actually provides the same answer as omniscience. So, a really surprising result there. We get the same answer as if we, in fact, knew everything. But what about this whole idea of, making the groups comparable or balancing baseline characteristics? So this is commonly used as the rationale for randomization. And again, randomization does tend to balance these things, but that's not the real reason for randomization. And surprisingly, once we've randomized, the baseline covariates actually have nothing to do with the validity of our conclusions. And in fact, specifically, if we end up with certain chance imbalances, that does not invalidate the trial, contrary to what some people may believe and what common sense may tell you. Covariate balance information can, however, lead to more efficient trials, so that would be in the form of perhaps fewer subjects. So in summary about randomization, the true purpose is not to provide balance in baseline characteristics between groups, but rather to provide a basis by which the groups can be causally compared, where the statistical results can be aligned with the scientific question of interest. Now, when it comes right down to it, how do you actually randomize? You just wait for each patient to come in and then flip a coin? Sure, you could do that. You could say heads, they get treatment A, tails, they get treatment B. It can work, but there are some problems with that. What if the FDA or another organization decides to audit? Or what if you're required to provide proof of your methods and how you track those things? It gets a little bit more difficult. Plus, if you're flipping a coin, it is possible to get, say, 30 heads and 10 tails out of 40 tries, which means you're not assigning as many patients to one group as to the other group, and things can get a little bit difficult in that scenario. And when you start looking at data early on, there could really be problems like nobody has been assigned to one of the groups, or very few patients. So that complicates the interim analysis. Well, one solution for some of these problems is something called permuted block randomization, which is the most common randomization method. An example of that would be for every 2 patients who come into a trial, you randomly pick either treatment A, then treatment B for those 2 patients, or treatment B, then treatment A. Or you could make those block sizes even longer, so for every 4 patients who come in, you randomly pick a sequence of treatment assignments like A, B, B, or one of the other 5 possibilities for assignment of those 4, and then you do the same for the next 4 with a different sequence, etc. Now, in these cases, if you use smaller block sizes, you're going to get better balance. But those smaller block sizes can lead to more predictability, which is a particular problem if you have an unblinded study. So a good solution to the predictability issue is to use random block sizes, which means maybe you assign 4 of these, and then the next time you randomly pick, say, 6, and then assign a random sequence of 6 treatments, and then go on from there. And each time you jump to a new block, it could have a different size to it. There are other randomization methods that are not quite as common as permuted block randomization, and we won't go into these in detail, but you may run into those in the literature. So, for example, complete randomization, earn or biased coin randomization, minimization or other covariate adaptive randomization, and response adaptive randomization, which typically fall under the umbrella of adaptive design trials. Now let's talk about missing data. In almost every trial, you're going to find some degree of missing data. Now, when you have fewer patients with outcome data, that's going to reduce the trial's sample size in effect. However, that's not a really big issue. You could just have, more patients in your trial. But the real issue is that missingness can be non-random. And the key here is that non-random missingness will completely subvert randomization. So all the causal conclusions that we just talked about and the ability of randomization to provide that is gone. And it removes our ability to have those great valid conclusions. And also, there's no good way to test for non-random missingness. So this is really a problem. Now, many have proposed statistical methods for dealing with missing data. For example, you may have heard of multiple imputation, principal stratification, or inverse probability methods. The problem with all of these and other methods for dealing with missing data is that all these methods make assumptions that cannot be tested. So the results cannot be interpreted causally. So the only solution to all of this is don't have any missing data. Now, in some cases, this is extremely difficult or impossible, but you want to minimize as much as possible any missing data. Another related topic is non-adherence. So some subjects don't adhere with their assigned treatment. And in the real world, we can't ethically force subjects to adhere to their assigned treatment. Now, going back a few minutes here, remember that I told you randomization allows us to be omniscient regarding the potential outcomes under different treatment assignments. Unfortunately, there is no way for us to recover the potential outcomes of, for example, what if the subject had adhered to treatment? This is because the adherence can, and very likely will, depend on the treatment the subject was assigned. For example, they're taking their drug and experience a side effect, so they stop taking the medication. Or maybe their disease got worse, which could have had to do with what treatment they were given, so they stopped their study medication. Or maybe they even switched to the other treatment, sometimes referred to as rescue therapy. Well, now we are really in trouble, because since randomization doesn't solve this problem with non-adherence, there is absolutely no way for us to recover the, quote, full adherence treatment effect. In other words, does the treatment work if you adhere to it? However, go back to the experiment. What we controlled for was the assignment to treatment, not adherence to treatment. That wasn't the experiment we did. Therefore, the only question we can really answer causally with the experiment we have done is whether the assignment to treatment works, not assignment plus adherence to treatment. Now, one proposed solution is the so-called per protocol analysis. So this means we analyze based on what subjects actually adhered to. Did they follow the protocol? So you throw out data for subjects who do not adhere. However, when you throw out data, you are inducing missing data. And we've already gone over this and said that missing data subvert the randomization and destroy the causal interpretation of the trial. So per protocol analysis doesn't solve the problem. The solution is to change the question that we're asking. And the question that we can answer is, what is the effect of assignment to treatment? We compare all subjects according to their assigned treatment groups, regardless of their adherence. This is known as the intention to treat analysis, and it's the gold standard of all trials, something that is expected to be done. Now, intention to treat can sound a little counterintuitive, and a lot of scientists object to it. And I feel that this may in part be due to some of the rationale that is typically given for intention to treat. For example, some say, well, intention to treat assesses the treatment strategy. In other words, the investigator intended to treat the subject. That's where the name comes from, which is, in my mind, a little bit misleading. Or sometimes they say intention to treat mirrors what would happen in real life. You know, in real life, there would be non-adherers. Well, again, these may be somewhat true, although I doubt that real life is mirrored very well by a clinical trial. But the real reason for intention to treat is that it is the only analytical approach that relies solely on randomization without additional assumptions, and that provides a valid causal interpretation. In other words, it is the only valid causal analysis we can perform. With this in mind, trialists cannot deliberately introduce missing data by excluding non-adherers, in other words, the per protocol analysis. And this also means that whoever is conducting the trial must make every effort to collect follow-up and outcome data on all patients, even if they withdraw from treatment. So again, the point being, withdrawal from treatment is not equivalent to withdrawal from data collection. I hope this module has been informative, and we will continue this in part 2 of randomized trials. Thank you.